Today, our speaker is our very own Dr. Sadie Ryan. Everybody knows her. I don't know if I need to introduce her at all, but given our shared taste in colors, if you been to our offices, you will know what I mean. So I volunteer myself to do this. So Dr. Ryan is an associate professor in our department, medical geography, and also at EPI, right? So she's the director of QDAC Lab Group and also co-director of Florida Climate Institute. Yeah, that's it. Okay.
That's it. That's great. Thank you so much. And it's really fun to be here today because I know some of you have seen some of this stuff over time, especially the faculty. Some of you have seen other bits of it over time. And some of you have no idea what I do, and you and I are new friends by virtue of this colloquium series. And so it's really exciting for me to do this today. I will try to get through all of it. I will pause in some spots and just check the time if somebody can like yell at me if I'm going over time. But hopefully I won't. And I seek to entertain. And so I just wanted to put a quick note on here. So it says by me and many others, all of my work is very collaborative. This is the result of a lot of interdisciplinary efforts across not just institutions, but also across agencies too. And so this is sort of a self-conscious journey talk about me sort of learning how to do some cool stuff. This is one part of a wide suite of research I do, but sort of trying to take this into the applied world has been a really interesting process, especially sitting inside a university. And so you'll see that come through today. So I want to start, or not. Let's get onto that. Okay. I want to start with a land and labor acknowledgement. So a lot of this work was conducted here, which is a crossroads of a great number of Native peoples, including the named tribes, the Timucua and the Seminole, and in an institution built in part using the labor of enslaved people. And I think it's really important to place ourselves in our context. A lot of what we're seeing today is the echoes of decisions past. And so when we start to talk about things like climate change, we tend to think that's off in the future. But in fact, the decisions we're making today are going to be those echoes in the future. And so when I say things like climate change, very specifically in this talk, I'm talking about what's warming up the planet. And so really straightforward descriptions like this show you the sun is shining onto the earth. There's a thing called radiative forcing, and that's the stuff that doesn't bounce back for a series of reasons, many of which can be attributed to man-made production of chemicals and things we do to the landscape. And so in amongst all of this is captured what happens to heating the planet. This is not a talk about climate change, this is a talk about the impacts of climate change. And so if any of you would like to dig deep with me into things like how you get to watts per meter, let's do that because I had to do that. This is part of doing interdisciplinary work. So as an overview for what I'm going to talk about today, I'm going to introduce you to a physiological model for malaria. You'll see what I mean by that. I'm going to show you some maps of what I call straightforward things, but they're not. And then I'm going to flip to Aedes transmitted disease models. Then I'm going to probably flip back and forth and confuse you between Aedes maps and more malaria maps. And then I'm going to talk a bit about vector-borne diseases generally and climate changes, models and decisions. And then I'll give you a quick zoom in to how this relates to things like decisions in Florida. And then give you a little summary on the utility and purpose of doing this sort of crazy adventure. So more than a decade ago, a bunch of us were sitting in Santa Barbara in this dream planet place called NCES, the National Center for Ecological Analysis and Synthesis, where a bunch of postdocs got together and wanted to ask this question: is a warmer world necessarily a sicker world? And we decided to focus on malaria. And so the first question we asked was, how does malaria respond to temperature? We're talking about a warming world. And so what we have here is a funky math equation, right? This is a McDonald sort of slightly adjusted equation for transmission of malaria. It's an R0 equation. You all know what R0 is at this point because COVID taught everyone how to be disease ecologists. What it is really is the number of secondary infections caused by a primary infection in a naive population. And in order to build this for malaria, we need a lot of malaria parameters that are about mosquitoes and transmission, and it's all very beautiful, right? So really what we're looking at here is we want to know about these mosquitoes and what they're doing. And so what they do is they suck up parasites from one person and the parasites develop inside the mosquito. They travel around in the mosquito and get into the gut and make this thing called an oocyst, right? And then the sporozoites bust out the oocyst and get to the salivary gland so it can inject the next person. Inside people, it goes and infects the liver stage. It gets into the red blood cell stage so it can start circulating again, gets to the next mosquito that bites it, right? All of this happens inside a human, where it's nice and temperately kept at your body temperature, but this stuff is happening in the environment. So this is what we care about when we're talking about climate change and malaria transmission, because the adult mosquitoes are flying around in the environment. And so what we have is these pieces that contribute to this transmission equation. And all of those pieces are basically made up of different bits of life history. And so we're all bags of chemicals. Temperature drives biological processes by speeding up chemical reactions. You start at some kind of critical minimum temperature where nothing's happening, it's too cold. Speeds up, these reactions speed up with all the chemical reactions, and you get to this optimal temperature where everything's just absolutely running perfectly. And after that, it breaks down really rapidly. This happens for things like feeding. This happens for things like laying eggs. This happens for the life history of mosquitoes. It also happens for the parasites inside them. And so you get this bump-shaped curve with temperature. So we said, what does that do to the cycled equation? Previously, people had measured this in sort of the temperatures they could see, and they thought these processes were linear, right? Temperature gets hotter, it all gets faster. And so we wanted to know, does it make a difference if it's a different shape? And so that's a math equation question. And so we went to the literature. We picked out all these studies where people had done temperature-controlled experiments in the lab. We fitted those responses with these curves, put them back in that R0 equation as a function of temperature, and got a curve out, and then said, can we validate this with field data? So to walk you through a year of doing that, these are the curves we got. They're all bump-shaped. They're all a little different. And so this is things like your vector competence, your survival, your parasite development rate, all those different components are all bump-shaped. They're not linear. So what happens when you put that in an equation for R0? You get a curve. Some things are pushing the curve one way, some things are pushing the curve another way, but you can get this optimal temperature for transmission based on using those parameters. You're all like, really? Just a blue curve? That's all you're talking to us about? Okay, so that was the previous estimate before we did it. That optimal temperature is about 6 degrees cooler when you include these curve parameters. When you look at 40 years of studies in the field, and you take the temperature from the location of each month which they're taken, or the entomological inoculation rate in the field, which scales linearly with R0. You don't need to know all that, but you do need to know where the data are sitting. So we were pretty excited about this result. So what do we do? We publish it, right? Put it in the Ecology Journal, big hit in ecology. And one of the words you'll see in the keywords is climate change. Is there climate change in this paper? No, there is not. But it has implications for climate change. So how do we show people this stuff, right? People like maps. And this is basically when I sort of switched over to thinking about how do we put this on maps? How do we talk to climate modelers? How do we start to think about this? Because the question of where you want to do intervention is really important for control. You want to think about prevention when you're traveling. You go to a travel doctor to get your shots and all your prep for going somewhere. They pull out a map, right? And they look, where's the place you're going? You also want to be able to anticipate transmissibility changes, right? Even without climate change. What season is it, right? Do you need your malaria meds if you're going there in January versus if you're going there in July? So what we did was with these first sets of curves, chopped up the quantiles of the curves and looked at the literature to see what people were saying to discuss how malaria looks on maps. Tried to match our mathematical modeling approach to that language, right? This is where we start walking into like, what are people saying in the literature? What is the agency language? And I pulled some world acclaimed temperature data. And this is our first attempt at trying to communicate this on a map. With malaria in Africa, there is aridity boundaries. So it can be too dry for mosquitoes to start existing. And so we use an NDVI product to mask out where it's too dry for malaria. And essentially what we're showing here is where it is too hot versus too cold and everything in the middle in each month of the year on average. And so we call this the sort of Goldilocks map, right? And so you can see that places like Southern Africa do get too cold under baseline temperatures for transmission. And that sort of fits with what people think they know. Ignore the stuff in the middle. It's a horrible visualization. We ditched it in future papers after this one. But it was an attempt to capture the whole landscape in one plot. If we take those older models with the older optimal transmission temperature, it looks like most of Africa is too cold for transmission most of the year. When we said that, people said, yes, but it wasn't supposed to be mapped. So that was my first hint that the world needs geographers. Because, oh boy, that's not a good answer. And so then I took what the climate scenarios were at the time and superimposed this on there and found some interesting stuff that where we took that very sort of highest transmission stuff, which is at the bottom, so that top quantile of most optimal transmission, we saw a decrease in the amount of area in the continent of Africa that would be suitable in that very optimal transmission. But if you look overall at all possible suitability for malaria transmission, that total area increases as we go through these climate change scenarios. So that's a weird sort of tingy little paradox there that is worth always thinking about when someone shows you a map of something under climate change, is what are you actually looking at? Because if we found it can go in both directions, depending which part of the curve you're looking at, it's worth knowing what those curves are doing. Okay, so then there was this question, sort of how do we really communicate this? And people like to know, for making decisions, how many people are you talking about? And so for this is a map of risk in terms of population and that sort of optimal suitability, thinking about where does that endemic zone move? So where is it sort of, up to 12 months of the year suitable in that optimal sort of top bits of temperature that are most suitable? And what does that look like as we move it across the continent, or sorry, as we move it through time in climate? And as you can see, this hotspot sort of bumps across the continent to land in very high density East Africa populations. This was also my first foray into population models, and really learning how to do population changes through time is something I had to learn after my postdoc. So you never stop learning, right? And so I've been sort of combining these, climate models and population models and having to do a whole lot of learning I wasn't trained in. And it's great because there's so many people who are excited about these separate pieces of disciplines that there's always someone you can talk to about it and be like, hey, what is currently considered the best product for this and why? The why is the really important bit. Okay, and so, we put this model out. Hey, we've, found this different thing happening, and it sort of hit the press. Some journalists interpreted it more or less correctly. Some of them didn't. And it happened to coincide with the Paris Convention on Climate Change. It takes a long time to publish papers. And so when journalists said, did you do that on purpose? I was like, welcome to my world. But, it was exciting, right? And so it was nice to get phone calls about that. And so it suggested to me that maps were, sort of a nice way to communicate some of these results, but that being really careful about the messaging with maps is something I was going to have to sort of bring with me. And so, we went back into our data mines. We started doing this for dengue, the same process to establish these thermal performance curves. And this time we had hit a particular moment in history where in the Americas, there were three arboviral pandemics three arboviral epidemics of the Americas happening, which were also in pandemic situations. So what we had at this point was two vectors, because there's Aedes albopictus as well as Aedes aegypti. Turns out they have slightly different temperature curves for transmission, which was a really interesting finding too. So these Aedes aegypti are a little hotter in their temperature curves, and the Aedes albopictus a little cooler. We had three different diseases happening simultaneously, and not everyone, but a lot of people were assuming that dengue was the best way to talk about this because viruses go really fast. And so if there are viruses going fast inside of mosquitoes, surely they do the same thing. Pin that thought. And we were now looking at a new continent and also a different kind of mosquito behavior, because these are urban dwelling container breeding kinds of mosquitoes. And so, I call this curve the great Zika and Dengue because it's got a little bit of everything in it. And we were validating this on case data, which is a really interesting thing to have to do. But that's what we had because people hadn't been looking at this in the field because it's new, right? And so the other thing we had done at this point was, because the data were a bit more sort of, hit and miss, we've been using Bayesian inference to try and hone down those parameter estimates. And so what we were using for our R-naught maps, instead of these normal, least squares curves that we'd had before, is we were looking at where we had this really high credibility, this posterior probability interval of greater than 97.5%. And so this is simply where R-naught can take off. So this is not saying it's greater than 1, it's saying where can establishment take place. And so in this case, what I'm showing you here is a map of how many months of the year the location is suitable for transmission by our Aedes albopictus, our cooler mosquito on the left, and Aedes aegypti, the hotter mosquito on the right. And you can see they look a little bit different, right? All right. I'm not sure about this sort of hemorrhagic map, but I got a little more into colors with time. At the same time we were doing this, because Zika had shown up, everybody was excited about it, wanted to know what was going on. There were a lot of global niche maps that occurred. So people were niche mapping the mosquitoes, they were niche mapping case data, they were niche mapping dengue and calling it Zika. There was all kinds of stuff going on, and there were lots of different maps trying to think about how we think about mosquitoes and spreading Zika. And one colleague took dengue cases and Zika cases, threw them both into the same model, and found that it looked different. So as I said, we're going to pin that thought about things being all the same. So we're at the height of this, I'm like, Zika all the way, Zika, 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 and then I got a phone call. So USAID had a contract. I got a call from this project called Atlas, which is looking at climate change. And they said, we read your malaria paper. We saw that the methods were very sort of transparently written. We want you to do that again with our climate models for our reports. And I said, sure. I didn't actually. What I said was, oh, it's really exciting. We could explore these different parameter spaces. And they said, no, we want one thing. And I was like, but there's other things that we could do that are one thing. What we want, we have a decision that will be made. And this is when I learned that sometimes decisions will be made, and you probably want to be at the table if your science is what you think needs to be at that table. And telling them you want to do lots of different things doesn't help. So bear that in mind as you start to talk to agencies and stuff like that about your science. Moreover, they had already named the report that I was going to put stuff in. And this is where I had like a little bit of contention with them, because I'm not modeling burdens. It is not case burdens. It is not malaria burden as described in the literature. What this is risk, right? This is stuff that could happen. And so I sort of wrangled the subtitle a little bit on the inside of the first page. So it's got a fuller title. So it's about malaria risk, but there we have it. And so what did I do for them? Okay, so USAID makes decisions on a regional basis. And so we have 4 regions, because we weren't considering Northern Africa in this project. That was not what I was asked for. And they have USAID colors, red and blue. And so what we have here is endemic and seasonal transmission as modeled by our thermal performance curves with their climate modeling. And so we looked at their definitions previously about what they were calling seasonal and endemic. So seasonal was 7 to 9 months, endemic was 10 to 12 months. So this is already showing you there's like different definitions in the literature of what people are taking these intervention actions based on. And so our results are going to look slightly different in different places depending on that. That was a fun one to realize. The other thing is when you look at this, you think, oh gosh, look at all that endemic area. Surely there's more of that. And this was a lesson I had to sort of teach people over and over again in the room, is that when you look at the people at risk, this thing called PAR, people at risk, people on the landscape are not evenly distributed. And so sort of density and area is a really interesting question to have to keep conveying to people. And so we see that the different regions have different numbers of people at risk in these different categories. I don't put price labels on people, but they do. And so people at risk is a currency they really wanted to know about. So it was how we could communicate between us, which was useful to find out. So when we look at this under these different climate change scenarios, so RCP 4.5 is a sort of slightly optimistic scenario, and RCP 8.5 is our sort of worst case scenario, also known as business as usual, depends which room you're in. So what we see for this different region is that as we push that forward through time with these climate models, what we see in Western Africa is a sudden drop off in the number of people at risk in this endemic areas. And what is that about? It's too hot for transmission. Pretty weird, right? It gets too hot for transmission. So a lot of that area ceases to be as suitable for transmission. What we see instead is we see that uptick in Eastern Africa, which is consistent with that finding we saw before of that high density hotspot moving across to land up in Eastern Africa. And we start to see things happening in Southern Africa. We see sort of two different scenarios in Central Africa, essentially, depending whether we're doing much worse on climate change or slightly better. And that's, another one of those interesting paradoxes that we have to think about as we think about how to tackle these things. When we look at the seasonal risk, I put that dash because that is an axis that's like another 40 million people bigger. So it's taller again than the previous one. And we see similar looking patterns as we move forward. And so this means there are different implications in the different regions that decisions have to be made. This is sort of, a very straightforward, nice USAID colors way of sort of describing how this is going to impact things. Another thing they wanted to know was if we are going to have new areas that will have endemic risk, where are those going to be? And so another really delightful colored map. It's really hard to depict this stuff usefully in reports, but we got there. And so as you can see, there are these patches of sort of brand new areas that are expected to be almost year-round suitable for malaria transmission in terms of climate. And so what does that mean in terms of new amounts of people at risk? We see the biggest uptick in Eastern Africa, as we might expect. We see a little bit in Southern Africa and a little bit in Central Africa. And so we see these sort of regional novel impacts emerging under these climate change scenarios. And that is the kind of thing that agencies can say, hey, that's where we're going to put some money for intervention. So it's really interesting to sort of take these maps into these spaces and figure out what that language is for talking about this. And so I'm just going to give you a quick list of lessons learned from doing contract work. Contracts in universities are very complicated. It's a lot of paperwork. Report writing is very different from scientific articles, but the need for clear communication is the same. You can't just be like, don't worry about the maths, right? You have to like lay out what did you do with this model and how did you link it to this climate model? The number of people, as I keep saying, or dollars are the basis of decisions. And so that should be the sort of metrics that are reported. And thinking about what scale the decisions are made really matters for what you're talking about. So in this case, it was regional, right? These are regional decisions by a giant global agency. The bits that sort of alarmed me, that it was really large financial decisions at stake. And here I was, sort of handing over maps. There's also multiple partners involved in lots of levels, and you may not know at the outset who you end up talking to. And that was a really interesting sort of journey for me. Sort of showing up in DC and being in a room of people and being told later who those people were was shocking to me, but kind of fun. It's a really great opportunity to learn, right? Being in that space was nerve-wracking, but also it's really a great opportunity to learn who does what and what they think about their jobs and what their motivations are. But as I said at the beginning of this little vignette, decisions are going to be made. And so being at the table matters. You can't complain about what models they use after the fact. Nobody cares, right? So you gotta show up. Anyway, in the meantime, we took our dengue maps and made some global maps about what we're going to see under climate change. So here we have the rainbow version of the months of the year that are suitable under these different scenarios. And current is baseline. So it's basically backing up to the beginning of the century, not actually current. And then we looked at what happens in sort of 2050 under RCP 2.6, which is now considered a bit too optimistic a scenario for the future, and that worst case or business as usual scenario. And what you see is they've been flipped a bit, right? So Aedes aegypti, our hot mosquito, is on the left, and Aedes albopictus is on the right. And what you see is as you sort of get into the worst case scenario in the future, for Aedes albopictus, you kind of see a crumbling of those like highly suitable numbers of months. And so you sort of see these two different mosquito physiologies really impacting what we're going to see these future predictions looking like. And so having done that, people like these rainbow maps. It turned out to be something that people were like visually tuned into. It's not the best way of doing mapping even, but it was, sort of nice to know that we had this image that would pull people in. a lesson I learned there is that the tables that journalists wanted to talk about, the journal articles don't want in the main text. And so we'd done this thing of sort of splitting it up by region, which I'll get into a tiny bit, but it was really interesting to figure out sort of what works for journals and what works for talking to people about what you've published. An important thing I mentioned in passing about Aedes mosquitoes is that they're container breeders. So when we started thinking about sort of people at risk, we're really talking about people living in built environments in the urban areas. And it turns out that when you sort of zoom into that urban area, this is work we did in Ecuador ages ago, but when we zoomed into looking at sub-city effects, we were trying to pick out, is rainfall driving mosquitoes? And it turns out that it's people's water storage behavior that is really driving that, and that we could find in some of these areas that it would sort of drown out the signal of precipitation. And so it became less useful to think about sort of precipitation as a climate driver when we're trying to look at these large scale sort of future scenarios, because it really is where people are. And that's sort of, the granularity at which we can get at this. So yeah, so then we looked at how many billions of people were going to be at risk under these models, right? So what you see here is we've got a huge chunk of the world is susceptible for 1 to 2 months of suitability. What does that mean, right? How much risk is that? I can't actually tell you. I can tell you that for 1 to 2 months of the year, the places that are looking blue on this map are thermally suitable for transmission to occur. Is that enough time for it to get established and get going? I don't know, but the future may bear out sort of us knowing a bit more about that as we pick up on where cases are. What those really sort of marginal areas tell us, though, is where we should be thinking about sort of doing a bit more surveillance, checking out which mosquitoes are there, checking what pathogens might be there, having people just sort of be alert that that is a possibility, whether that's point of care training, whether that's public messaging. But it's good to know sort of where those boundaries of these ranges are, as well as sort of where the core transmission risk is, right? And so what we did when we looked at this, when we wanted to put this on a sort of climate change schedule, as it were, is we looked at multiple global circulation models. So that is the height of the box that you're seeing is the variability between models. And then what you're seeing at the bottom is marching across the different concentration pathway scenarios presented from 2.6 up to 8.5. And then on the left-hand side of your screen, you have 1 or more months of risk. So that's anything from the sort of dark blue up in the previous map. And then the right is year-round transmission. So that's where do you get endemic transmission. And what you see is the left set of panels is our hot mosquito, and the right panel is our cooler mosquito. And what you start to see, if you sort of cruise all the way to the right of the screen, is that it gets too hot for albopictus to keep sustaining transmission under these future scenarios. So this gives us a nice mixed message, right? Because all the other ones are saying things are going to get worse. Are we going to end up in an Aedes transmitting world? Are we going to end up in a less albopictus transmitting world? It's hard to know, right? And so these are sort of ways of thinking about how to think about this. One thing that's pretty clear is that the variability due to the different global circulation models, so these different atmospheric physical based projections of what the climate's going to do, they're kind of chunky in our sort of any transmission. So we sort of see the width of that spread out a little bit, suggesting those margins might be different under those different models. But what we see is pretty tight scenarios in the future for that 12 months of transmission risk. And so across all of those RCPs, Aedes aegypti is going to do a wee bit better at transmitting to us. So moving on from there, I mentioned this whole thing about tables and appendices. This looks really boring, right? But so what we did is we took this global burden of disease regions approach. These are regions that are used in health assessments across the world. And we were able to sort of classify which of these regions are going to have more and less risks. And we did this in terms of people exposed for the first time to any transmission risk in these future scenarios. And so this says how many new people are at risk, not simply adding to what's already there, but how many new people are we going to get? It turns out it's kind of a lot. And it turns out that which region is worse affected changes depending which vector you're looking at, so which Aedes mosquito you're looking at. All right, so we're going to move on from our table there. And so what we did is we then went back and we said, okay, what's happening in the places we've known about malaria? We know that dengue might show up more in parts of Africa as climate changes, and as we sort of recognize that it's actually a big part of the urban population febrile disease makeup. And we looked at data that a colleague had where it's like all causes febrile diseases coming into clinics. And it was those that tested that weren't malaria that seemed to show up at dengue temperatures, is your takeaway from this model. And basically we were able to look at sort of what happens when you compare the difference on the top row between this malaria hotspot sort of moving across the continent. This is new climate models, right? We've moved forward years in time. Climate models have upgraded, but the maps look the same because I wanted them to look the same. And so we're still seeing this hotspot doing the same sort of march with populations ending up over in East Africa. But when we do the same thing with dengue, we see this sort of spreading of that hotspot. It kind of spreads right across the middle of the continent. And so we're getting this sort of increasing potential risk for dengue transmission, which suggests there may be this shift, right, from malaria to dengue fevers as a primary concern. And so from this, we've sort of said, okay, we've gone from like these absolutely unintelligible mapping things, right, to something that's slightly intelligible. And hopefully we can translate some of the mapping approaches into these actionable risks by using things like PAR, right? There's people at risk, and that helps with policy decisions. But we're still getting these sort of like, shock and awe headlines going on here, which it's frustrating to me when, things like The Guardian are like, there are these mosquitoes coming from Florida. And I'm like, oh, that's not what I said, And so, sort of figuring out how to sort of pitch this stuff is really complicated because climate, people are scared of climate change. And when you try to talk about this, you need to not overstate what you're talking about, but particularly journalists need a good story. And so trying to sort of capture this in sort of the right kinds of maps and talk about the right kinds of actions from those maps is important. And of course, as you all know, because you're geography people sitting in a room, a lot of that's about scale. But people tend to switch off when you start telling journalists about issues at scale. So remember I said, hold on to the thought about Zika. So colleagues went into the lab and they infected Aedes aegypti, same mosquitoes, with dengue and Zika and ran them in parallel through these temperature experiments and came up with curves about this thermal performance of transmission and said, hey, look, the outcome is the same, right? And I was like, hey, look, something weird's going on down at these lower temperatures. So I thought, I'd like to see that on a map. And if we pull apart where the parameter space is, vector competence, the ability of the mosquito to replicate and then transmit onward, is set at a slightly higher temperature for Zika than for dengue. And we see that sort of that the two colored curves kind of echo that same slight temperature difference at the bottom end. And so if we do that on a map, it does look quite different. And so on the leftmost one, we have Zika. And in the middle one, we have dengue. And then on the very far right, we have the two of those kind of superimposed on each other. And so I said, yikes, because that is quite different. And we had assumed that dengue and Zika were probably doing the same thing, right? And so we need to think about this fact that there are these very, very exquisitely unique relationships between pathogens and vectors and temperature, that it's, you sort of need to get in on that experimental level to understand really what's going on with this. That not all, I'm going to say not all dengues are the same either. There's actually 4 potential co-circulating dengue strains. And so as we start to sort of, push this effort forward, I don't hang out in a lab putting mosquitoes through these trials, but I have lots of colleagues who do. There is so much work in this realm to be done yet. And so, getting these thermal performance curves and thinking about the climate implications, there's just years of that to go. And I'm so glad I know people who are like really into it, right? So we saw that Zika is similar, but different. It's still Aedes. It's a slightly different temperature profile. And one thing that you may not have noticed, but has happened, is we've stopped talking about Zika, right? Where did it go? There is apparently this large-scale population immunity, but we do need to think about sort of novel exposure. What happens when Zika shows up again in a generation when people haven't been exposed to it, if they haven't? What will that mean? What will it look like? So if it's the same Zika, which research has been done since to suggest there's lots of Zikas. But if we think about this in the climate context, we push this forward one generation to 2050, right? And looked at these different climate scenarios. What happens if it pops up again in the world? Large scale. And so this is our baseline sort of, I say current, but it's really baseline thermal suitability limits, and those numbers of months again. Sorry, my voice is really cracking here. We estimated that about 5 billion people are exposed to 1 or more months of potential risk, right? So that's like, where can it show up and happen for 1 or more months? If we look at 2050, it kind of moves. It does the poleward shifts that we would expect under climate change. And so we looked at these sort of 4.5, 8.5 scenarios again. And then we did the global burden of disease risk assessment on that to say, where will we see different regions popping up under these 2 different scenarios? And so this is really asking sort of indirectly a question about mitigation or not, and sort of what's going to happen. And so if we think of it as prioritizing regions under this global burden of disease question, under 4.5, so with mitigation, high income North America gets the biggest new increases in exposures. Without mitigation, we see this Eastern Sub-Saharan Africa pattern popping up again, and that gets the biggest increase in new exposures, but that's all relative, right? Under 8.5, a lot more people are at risk. And so we sort of need to think about, when you read policy documents where they're like regionally, blah, 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 and so these decisions must be made, think about sort of the relative impacts of thinking about regional priorities of what that's going to mean. Yeah, I was a little worried when we sort of, came up with this first set of results. I mean, the results are the results, right? Like, they are a bunch of models, crank the handles, get the answers. And if they're not answers you like, it's still the answers, right? So sort of, saying with mitigation, we're going to be worse off here is not actually what this is saying. It's just a prioritization thing. So, okay. So there I was, pandemic struck, lockdown happened. I was stuck in my house in Florida. And I said, well, what about Florida? People are always asking me like, hey, can you show me Florida on that map? And I'm like zooming in, I'm like, it's a bit ridiculous. And so I found some climate models that were better resolved just for North America. So sort of calibrated to us sort of here, including parts of Canada. And I, really, really good small resolution, 1 kilometer squared. And so I also got one that had a baseline that was moved up a little bit. So it wasn't the very beginning of the century. It was sort of calibrated on 81 to 2010. And so I took those monthly means again and did these, sort of took these futures from that model set. Again, the same scenarios I love, the 4.5, 8.5 comparison. And I wanted to check out dengue, Zika, and West Nile virus, because we now had thermal curves for these 2. And I was curious about Florida's population. Florida's population is increasingly urbanizing. People are moving here. People will keep moving here. We had a 2015 estimate of 20 million people, 2021 estimate 22 million. So that's like a steady increase, right? Going to keep happening. And people have noticed that where you get increasing urbanization in southern Florida, it creates what's called ecological filtering. They didn't call it that in their publications, as me imposing my training on them. But basically it boils down to, you get these very urban happy anthropophilic, so they really like biting people, culex and Aedes aegypti mosquitoes. And basically the rest of the mosquito community is just lost as you get more urban. And so it's really focusing down to these like key disease transmitters. And so for me, that's, it's fascinating, right? But it's also troubling. And so I thought, sort of, how can we bring this framework together? We also published a paper that there was a dengue outbreak going on during COVID, and there was concurrent dengue and West Nile, and they were picking it up in the mosquitoes, and it was a different dengue strain than people were transmitting amongst them. And so we had sort of got this evidence that stuff still happens even when we're all, hanging out in our houses. And so I was really curious about, this place I'm living called Florida. And so I took some of these models and squashed them down. And if we look at dengue at a baseline for Florida, this is what that looks like in terms of the months of suitability, because as you'd expect, it's all quite suitable. But so we're sort of floating in that 4 to 9 month range for suitability for dengue, right? Nobody's particularly surprised by that, right? When Zika showed up, where did it show up? So at that baseline, it looks colder, right? These nice colder colors. That's still sort of in the 4, 5, 6 range, like number of months a year. That's still half the year where we had 23 counties that were suitable for 6 or more months. And that was a population of about 12 million at that point. You can see where this is all going, right? You can see that I'm having these like stacks of papers building up that I need to write, where we just crank the handle on the population's temperatures and just keep going, producing all these maps. But so, that's what it looks like. If we take this in the future, we see that 6-month line go up to the border with Georgia, right? So we're seeing that happening. But we also have this thing called West Nile virus. West Nile behaves a little bit differently. It's a spillover disease from birds to us, mostly through mosquitoes. And urban birds are doing interesting things. We like urban parks. We should explore what we're doing. It has horrible neurological implications. It's here. It's been here. It's going to keep being here. What does that look like to you? Dengue or Zika? Yeah, right? So we've got this other disease that's co-circulating, that it has slightly different dynamics, that it's a spillover. We don't pass it to each other. We are dead-end hosts. But it is still here in the same way that dengue is here. These are urban mosquitoes. Vector control can be similar. And so what does this all mean? Like, what am I doing noodling with these maps and showing them to you, right? So what it means is that vector control burden increases. Vector control is funded differently in different places throughout the state. Sometimes it's your city taxes, sometimes it's state money getting into local taxes, sometimes it's a region, right? And so there are these things called vector control districts. They all serve a different amount of people in different ways. They have to hire something called seasonal workers, right? What is seasonal? How long is that? If you have to hire people for more than a season because your susceptibility is all the way up at 10 to 12 months. What does that mean for your budget, right? Like, these are budget decisions that need to be made. So we need to think about how we anticipate these shifting boundaries on risk. We need to add surveillance and screening. I'm going to say that for everything all over the world, but if we can start knowing before it comes, that's really helpful because we can prep. We need more of that point of care training. So, clinicians, clinics, like information that can get into the hands of people and clinicians. We don't really have anything other than palliative care for things like dengue and West Nile. There's no vaccines available yet, widely spread. We're really good at making habitat for mosquitoes. I'm sorry about that. Everybody knows about tip and toss, right? We all heard a lot of that, especially when Zika was the thing we were most worried about. But as soon as COVID happened and everybody went into lockdown, they started going on walks. I'm sure they forgot to put their long pants on and put their socks over their long pants and wear their mosquito control and tip and toss. And so this is going to keep being, something that pops up again. We're going to keep having arboviral risk here. So where are we going next with this too? Yeah, I can do this. Okay, so there's a new mosquito on the move. It's called Anopheles stevensi. It is a malaria transmitting mosquito that has its original range sort of in the Indian subcontinent and throughout parts of the Middle East. It has started moving into Africa. There's an urban form of it that has gone particularly well into parts of Africa and established. And so we keep seeing these reports of this happening. The WHO is like, this is the most important thing right now. Let's think about where it's going. And we had a temperature model of the mosquito itself a few years ago. We put that out with maps in its native range. And when we compare that to a niche model in its historic range, it turns out that temperature better predicts where it's going to sort of establish. So this is some work looking at sort of novel establishment sites in prep. Got to move on it sometime. And so we've been thinking about sort of how do we take our thermal performance curve measures and sort of rerun all that math with stevensi involved in this. And so some colleagues and I sort of got this out just at the end of last summer. with separating out the different anopheles, Stevensii and Gambier curves with 5-axis and different malaria, because they talked about that exquisite relationship between vectors and their pathogens and why that matters. And so we've teased that out a bit. Shri, who's just finished up her PhD, looked at some of this for what it might imply for climate change in Nepal. So that's this nice map here that we might see, Stevensii may establish and sort of be part of the ongoing issues with climate change and malaria shifts. And then we've got some global maps. We just sent back the revision on that yesterday. We're doing, cranking the handle, trying to do this again, think about the people at risk. And of course, what's happened in between times is IPCC happened again. A lot of our stuff ended up in there as like supporting parts of many chapters. But we now have a bunch of new models. We've got all the CMIP6 scenarios to think about, and to think about sort of how those different scenarios relate to our views of sort of ecological stability in those models themselves, and how that will then work with some of our vector-borne disease work. So crank the handles, more papers, right? Isn't that the name of the game? But it's sort of, it's fun to keep doing that. It's really nice to be productive. It's really fun to get students involved in thinking about how we even think about this kind of modeling and communication with maps. But it is worth thinking about what this is, right? And I think it's really important. People sort of say to me, gosh, you work in climate change, isn't that horribly depressing? And I say, well, I came from conservation. So, I'm used to it, but I really think it's useful to think about this in terms of planning tools. The great thing about vector-borne diseases is we actually do have interventions. We've had them for a long time, so we have the toolbox. So we know that what we need to do is know where to do what and when, right? And so that's why this is sort of, it's compelling to me. It's what makes me keep working on this so hard. When I said decisions will be made, they will. People have to make decisions. It's public health. People are going to make decisions. We need to really target that on-the-ground surveillance where it is and think about how we can respond to things like changing epidemiology as things change, and sort of what that's going to mean for people on the ground, right? Like boots on the ground. When we're talking about things like USAID money, thinking about how you can improve a country's capacity for collecting and using information at a national level should be part of a region and sort of, have everyone involved. Building capacity in health systems is always going to be a weak link, but we need to think about it. I don't particularly like helping people refine budget strategies for international programs, because country selection is a horrible word, but it is what it is. How do you prioritize if you have one budget is an important thing to think about. And we want to keep developing these regional approaches and partnerships. And so, some of this stuff has ended up in that IPCC report, as I mentioned. Simply building an evidence base for what we believe is going to happen under these different climate change scenarios, and having that out there helps the global community think about it more. Since we are a geography audience, I want to talk a little bit about the scales of questions and the utilities of these maps. This is like my limitations slide, shall we say. There's a whole distribution of issues when you're playing with like global mapping, right? I pull down rasters that are global rasters, right? That is definitely going to have different uncertainty at different scales. So if you scale it down, you're going to get data uncertainty that is either a mismatch between local and global phenomena, or it's simply that you've scaled it down too much. You also get data uncertainty when you take a model that was based locally and scale it up, right? So scaling problems go both ways. We also have massive data uncertainty of the future, because we talk about climate model outputs as data. And what we know is it's a physical model, and so it's grounded in these wonderful physical relationships, atmospheric models. But you got to remember there's lots of them. Got to remember that they do different things in different regions of the world, so no one model is the perfect one. And so we also don't know that we're going to end up exactly on the path that these models represent. So that's the sort of data uncertainty of the future to think about. We also inform ourselves as we go, and that's a whole sort of ontological problem I'm not going to get into. The key thing is intervention tends to be local-ish, right? If you think about vector control, spray teams, spraying for mosquitoes, that's very local, right? And a global model tells big policymakers something, but it doesn't necessarily tell my spray team where to go next week. And so thinking about sort of how you go between these scales is really important. Regional scale planning and policy tends to be how this stuff is handed down. And so that's, sort of me trying to get into that policy sphere such as I can. So these sort of mesoscale maps really kind of matter. Importantly, sharing data, sharing code, sharing all of this stuff out freely and openly is absolutely important to being able to do this stuff, especially partnering with international public health organizations, with companies who aren't necessarily inside academia. We put all the rasters and code online. It's just what we do. Publish it, it's out there. Have it, take it, mash it up, make it better. Argue with it, please. Yeah, so I'm done, but I wanted to thank my funders and collaborators. We've been funded through a lot of different things. I don't know if everyone in the room knows, I'm 50% joint appointed with the Emerging Pathogens Institute, and I bring in a lot of my salary off external grants. And so I'm sort of spread across lots of things. I wear about 7 different hats every day. And I'm currently part of something like $34 million in grant funded research ongoing. And that is a lot. So if you see me running around and being on Zoom all the time, it's not because I like Zoom, okay? But please do come and chat to me. I really love talking about this stuff. I love talking about anything pretty much. But so with that, I'll thank you and ask if you have any questions. Yes.
Every time I see one of these talks, I get all confused about which mosquito carries which pathogen. But the question is there any kind of interactions amongst the different mosquito species that might have that we know about?
Yeah. So one thing to sort of bear in mind with these, I'm going to pick on the Aedes mosquitoes, even though I've talked about other things invading next. So Aedes mosquitoes aren't from here, but they're here, right? And so we know that when Aedes aegypti shows up, it establishes. We know that when Aedes albopictus comes in, it may or may not displace them. There's weird larval interactions that happen. Like the larvae themselves do different things and can like predate each other. And so then you get in these like arms races about which one can develop fastest and get bigger faster than the other one if they're in the same habitat. But the sort of the mechanisms of that are, those stuff of great behavioral science, right? But so thinking about, what establishment pathways even look like and why they are what they are is fascinating. And we keep changing the food on the landscape. One thing to remember with mosquitoes is we are blood meals, but also we put animals on the landscape that are also blood meals. And so we can shift the composition of all these things. And then it all comes back to us. I hope that answered the question.
Well, it does. I'm going to have people some references, but not now.
Yeah. Absolutely. Any other questions? You're all like, what on earth was that?
How quickly can the mosquitoes adapt?
That is a great question, and one that I really love, like asking mosquito people about particularly. So there are hard boundaries on how much you can sort of shift metabolic adaptations. And sort of that lethal limit is pretty hard according to mosquito people, but there really is like an upper limit to how hot a mosquito can be. But if you look at, so I'm not going to flip back and forth, Anopheles stevensii are really hot mosquitoes, like they are super hot adapted. And so they can go, their temperature curve is really shifted over when we sort of do the math. But obviously it's always going to be pushed back a little bit by that lethal upper limit. But yeah, the question is also like doubly complicated because the pathogens can also sort of shift their thermal tolerance. And so the question is the mosquito and the pathogen together and how fast they can evolve together. And I have colleagues who are looking at that in the lab for West Nile. Super cool. Yeah.
Yeah, after the Civil War, John Muir hiked all the way to Cedar Key, 1,000 miles. Caught malaria there. When did they get rid of malaria then for?
What's that?
When did they finally get rid of malaria?
So I don't know when they got rid of it exactly. I know that malaria has sort of locally established a couple of times in the US in the past like decade or so even. But that's usually sort of travel introductions, people coming back with it. And so it's always possible for it to be here, but they did a heck of a lot of like landscape modification was really a key way to get rid of malaria mosquitoes. I know that there was I'm going to say a date and don't quote me on it, but in the 30s in New York City, there were heroin dealers lacing their heroin with quinine because there was still, potential malaria happening with people spreading it with needles as well as there being a small amount of mosquito activity. And so, yeah, it is both very recent history and very possible to happen again if we're not sort of keeping our eyes out.
There was a significant bunch of malaria in southwestern Georgia recently, in the 50s.
Yeah.
That's when Bob Woodruff came into the Jones Center, the CDC was established.
Yep.
Question
The
from
introduction.
online.
Florida was a frontier until the 30s, so.
Did somebody online say they had a question?
Yes, it's Esther.
Yes.
I was just bouncing off of this is really interesting. Thank you. And I'm sorry, I kind of dropped in a bit late. But I'm wondering about, and I know nothing at all, but it's based off of what you said about the temperature tolerance of these mosquitoes. Is there any possibility that they can adapt?
Yes. So,
Okay.
so Brian asked about the sort of evolution side of it. There's also a behavioral side of it where mosquitoes are really smart. They can find the shade, right? So when you talk about temperature, and we're usually thinking about sort of, the air temperature at a certain height of a man is sort of what we think our temperature's measuring. The mosquitoes can always find somewhere nice and cool to sit. And so they can behaviorally avoid temperature extremes, which is, it's not really why I use temperature averages, but it is an argument for not sort of using the extreme highs and lows of the daily temperature measurement. Yeah. Smart guy. All right. Is that it?